In this lecture, I'm going to talk about effective lines of innate defense that interfere with establishment of infection, and if a pathogen is to establish an infection in a susceptible host, actually a series of coordinated events must circumvent both innate and adaptive immunity. So if innate defenses are overwhelmed, the second line defense are mobilized to ensure host survival. I mean by the second line, these comprise the adaptive immune responses. So the effective lines of innate defense that block establishment of infections, they are so many, but I'm going to focus on very important of them. For example, the anatomic barrier of the innate immune system. And actually the innate immunity is the definition of innate immunity is a basic immunity that we born with. And actually we born surrounded by 4 barriers. One of them is anatomic barrier. Anatomic barrier is skin from outside and mucous membrane and mucus from inside. And as you know that the pH of the skin is between 3 and 5, and this pH is not suitable for microorganisms to grow. And also in case of mucus is very slippery to pathogens, and it has or it contains some antibacterial and chemicals that kill the pathogen. The second barrier that we born with is a physiological barrier. So one of the physiological barriers we have is hydrochloric acid in the stomach, and also when someone gets infected by a virus, for example hepatitis B or viral hepatitis B, so the infected cell will produce antiviral, for example interferons, and just to protect adjacent uninfected cells. And also within the gut there are specialized cells that produce antibacterial peptides. So as I mentioned, that production of interferons by viral infected cells protect uninfected cell, and this is actually very effective line of innate defense. And also the presence of the normal gut flora, also it is one of the lines of innate defense. And the cell walls of some gram-positive bacteria contain a peptidoglycan, and that, as you know, that peptidoglycan activates the alternative pathway of the complement, and this results in generation of C3b, which opsonizes bacteria and enhances phagocytosis, as can be seen here. This is phagocytic cell, and this is gram-positive bacteria. They produce the peptidoglycan, and peptidoglycan will activate the alternative pathway, and accordingly this results in formation of C3b, and C3b will bind to the bacteria just to overcome the negativity on the surface of the phagocytic cell, and this actually helps in phagocytosis of the bacteria. And also, and here, as can be seen here, in macrophage, for example, there are receptors for C3b, and this process is known as opsonization. And also in dendritic cells, there are specific receptors for C3b. And one of the most effective lines of innate defense is toll-like receptors on the surface of phagocytic cell, and maybe macrophages, surface of the macrophages, and surface of the dendritic cell. And as can be seen here, this is a toll-like receptor for macrophage, and actually the discovery of these toll-like receptors paved the way towards understanding why some individuals may produce and respond better early to dangerous pathogens. So if someone gets infected by this bacteria, for example, and the bacteria will activate this macrophage. As activated, macrophage will produce cytokines, for example, tumor necrosis factor alpha, interleukin number 6, interleukin number 1. So some bacteria, they have lipopolysaccharides, LPS, and this LPS actually stimulate production of these very important cytokines by the macrophage. And as you know that tumor necrosis factor alpha is an inflammatory cytokine, and it is responsible for diverse range of signaling events within cells. So these events may lead to necrosis and apoptosis, and also important for resistance to infection by pathogen. Similarly, interleukin 1, it is essential for the host response and resistance to pathogen, and also it controls innate immune processes. So bacterial of LBS of gram-negative bacteria, this stimulate the production of cytokines by macrophages or endothelial cells, and stimulate release of very important cytokines that play very important role in an innate host defense. Let us now come to toll-like receptors. These are actual receptors expressed on the surface of some cells, for example, macrophages, dendritic cells, and other cells. And toll-like receptors, these are pattern recognition receptors, or PBR, and so they are well-defined pattern recognition receptors responsible for pathogen recognition and induction of innate immune responses. And as can be seen here, these are toll-like receptors on the surface of these cells, and they are known as pattern recognition receptors. So they recognize pathogen associated molecular patterns. As I mentioned that several pathogens, they have molecular patterns. For example, gram-negative, they have lipopolysaccharides. Gram-positive, they have peptidoglycan. These are molecular pattern. But also and these are known as PAMs. And also the pattern recognition receptors, they recognize dams or damage associated molecular pattern during some severe inflammatory disorders that lead to tissue damage. And one of the effective lines of innate defense is a process of phagocytosis, and actually this is a very important barrier. The phagocytic barrier is a very important barrier of the innate immune system. So phagocytosis of a pathogen or infected cell, as can be seen here, this cell is infected. This is actually red blood cell infected, so can be phagocytosed by macrophage or by monocyte and macrophage. So phagocytosis is very important effective line of defense. Natural killer cell also plays a very crucial role in the innate immunity. And as you know that natural killer cell, they recognize cells that express major histocompatibility class 1. For example, this host cell, when get infected, and also recognize host cell infected by viruses, and also recognize the NK cells, they recognize cancerous cell. So NK cells normally they have an inhibitory receptor on their surface which interact with major histocompatibility class 1. So this is an infected cell. But when the cell gets infected by a pathogen, and one of the strategy of the pathogen is to evade means to escape the immune response, so there will be downregulation of major histocompatibility class 1 induced by the pathogen. In this condition, this inhibitory receptor will not find the major histocompatibility class 1. So we recognize this cell as an enemy and produce perforin and granzymes. Actually, perforin will induce perforation of the surface of the infected cell, and granzyme will enter through these pores and cut the nucleus and induces what is so-called programmed cell death. So, but however, I find it will be interesting to this lecture also to highlight the failure of the innate defense or innate defense overwhelming. So the evasion means escape of innate immune responses by pathogens as a survival strategy. It is actually the pathogen may evade the innate immune responses just as a survival strategy. They follow several ways to evade or to escape immune responses. Pathogens may grow within host cell, not seen by the immune system. They will be sequestered or hidden inside the cell, so they will not be seen by the immune system. And as I mentioned here, that some pathogen, when they enter the cell, they downregulate expression of major histocompatibility class 1. And for example, viruses, when they enter, some viral protein will bind to major histocompatibility class 1, and this major histocompatibility class 1 will be expressed on the surface of the cell, and this is a human cell, actually self cell, and major histocompatibility class 1 when get expressed on the surface of the cell, also is expressed with or in association with viral antigen. So this viral antigen will be recognized as non-self, and this will produce cascade of events that end in killing of the virus. However, the viruses when they do that and down-regulated major histocompatibility class 1, this will be sensed by NK cell, and NK cell also has the capacity still to kill or to destruct the infected cell by the virus. So as I mentioned that these microorganisms, in order to evade effective immune and responses and neutralizing immune responses, they grow within the host, and maybe they get sequestered inside the host and will not be seen by the immune system, or they express some molecules very similar to those of host cells, so they will be seen by the immune system as a self cell, and or may cover themselves by plasma or by host membrane molecules. And in some instances, for example, in Epstein-Barr virus and other viruses, cytomegaloviruses, they produce interleukin 10-like. And as you know, that interleukin 10-like, interleukin 10 itself, it down-regulates macrophages, and as you know that macrophages, they produce human necrosis factor alpha and interleukin number 1, and also they produce interleukin number 12. Interleukin number 12 is very important for activation of T helper 1, and activated T helper 1 will produce interferon gamma, which it has antiviral activity. So some viruses, they produce some materials very similar to cytokines, for example, interleukin 10-like by Epstein-Barr virus, or in some instances, the pathogens may change the molecules on their surface, and this is known as antigenic variation. So when the pathogen is under attack by the immune system, it will change the gene segments, and then this will result in formation of new protein, and so the immune system will see this protein as if they see it for the first time, so it will produce a new defense against it, and then the pathogen will again change the surface molecule, and this actual process may exhaust the immune system, and also the immune system in such condition may produce some antibodies of low affinity. Thank you very much, Professor Ahmed Bola.